Good morning, dear student. In previous lecture, we already discussed what is enzymes and their classification. Okay, some important properties of the enzymes and their classification. So, in today's lecture, we are very much interested in the second important topic of our chapter, that is chemical nature and the catalytic activity of enzyme. Chemical nature and the catalytic activity of enzymes. So in this point, so let us discuss some important points regarding to these chemical natures and catalytic activity of enzymes. We know that all all enzymes are proteins. All enzymes are proteins. Okay, so we know that. So whatever the enzymes, they are nothing but proteins. All enzymes are proteins. The second important point is that only protein part, so only protein part, only protein part of enzyme, only protein part of enzyme, it is known as apoenzyme. It is known as apoenzyme. Okay, only protein part of the enzyme, it is known as apoenzyme. While if you consider the third point, that is non-protein part, non-protein part of enzyme, non-protein part of enzyme, it is known as coenzyme. Coenzyme. So you should keep in mind the two important points, that is we know that all enzymes are proteins. The protein part of the enzyme, it is known as apoenzymes, while the non-protein part of the enzyme is known as coenzyme. So when these apoenzymes and coenzymes, okay, combine together, it will gives hollow enzyme or hollow enzymes. Okay, so what is this hollow enzymes? Hollow enzymes is a combination. Hollow enzymes. Hollow enzymes is a combination of apoenzyme, apoenzyme plus coenzyme, apoenzyme plus coenzymes. So we know that. Okay, so the protein part of the enzyme, it is known as apoenzymes, while the non-protein part of the enzyme, it is known as coenzyme. And what is this hollow enzymes? So combination of both apoenzymes and coenzyme, it will forms hollow enzymes. Okay, and if you see, the next important point is that uh, neither apoenzymes or nor coenzyme is catalytically active. So you should keep in mind. Okay, that is neither apoenzyme, neither apoenzyme nor coenzyme is catalytically active. Catalytically active. Okay, so you should keep in mind the combination of both apoenzymes and coenzyme, it will forms hollow enzymes. Neither apoenzymes or nor coenzyme is catalytically active. Only the hollow enzyme is catalytically active, and it will involve in the conversion of substrate into the product. So only this hollow enzyme, it is responsible for conversion of this substrate molecules into the product formation. Okay, only the hollow enzyme, it is convert the substrate molecules into the product. So neither apoenzyme individually, neither apoenzymes nor coenzyme is catalytically active. So if both are present, some are protein part, some are non-protein part, then it is responsible for the conversion of substrate into the product. Then it is responsible for the conversion of substrate into the product. So let us discuss the next important points regarding to this chemical nature and catalytic activity of the enzyme. So this haloenzyme, it is a combination of apoenzymes and coenzymes. Okay, the only protein part, uh, the protein part, it is known as apoenzymes, while the non-protein part, it is known as coenzyme. The non-protein part, it is known as coenzyme, and this coenzyme or the non-protein part, so you should keep in mind that is non-protein part, non-protein part of enzyme means your coenzyme. Okay, non-protein part of enzyme is responsible, is responsible is responsible for imparting or increasing the catalytic activity of the enzyme, responsible for imparting the catalytic activity catalytic activity of the enzyme. Okay, so this non-protein part, it is known as coenzyme, which is responsible for imparting the catalytic activity of the enzyme. So if it is present, the non-protein part is present, okay, coenzyme it is present, then it is responsible for increasing the catalytic activity of the enzyme, and it is also known as this non-protein part, it is also known as cofactor. It is also known as cofactor, so you should keep in mind. So what is this coenzyme? This coenzyme is your non-protein part, and it is responsible to increasing the catalytic activity or imparting the catalytic activity of the enzyme, and it is also known as cofactor. Okay, it is also known as cofactor, and these cofactors are divided into three types. Okay, this cofactors, if you consider this cofactor, there are three types of cofactors observed in the enzymes. Okay, so three types of the cofactor observed in the enzymes. This cofactor is nothing but your non-protein part of the enzyme, it is also known as coenzyme. So this cofactor I can classify it into three types. First, that is coenzyme. Okay, so we already discussed coenzyme. So we already discussed combination of apoenzyme and coenzyme. Coenzyme is a non-protein part, so it is also one of the type of this cofactor. Non-protein part, it is known as coenzyme. Second, that is your metal ion activator, metal ion activator, and third one, that is your prosthetic group, prosthetic group. Third one, that is your prosthetic group. So at least you should understand that all enzymes are protein, and the only protein part of the enzyme, it is known as apoenzymes, while the non-protein part is known as coenzyme. And when the combination, when these apoenzymes and coenzymes combine together, it will forms haloenzymes. Individually, they are not showing the catalytic activity. So neither apoenzymes nor coenzyme is catalytically active. Only the haloenzyme is catalytically active, and it converts the substrate molecule into the product. And if you consider the non-protein part of the enzyme, it impart the catalytic activity of the enzyme, and it is also known as cofactor. These cofactors are again classified into three types. Okay, so there is a three types of cofactor observed in enzyme. One is coenzyme, second is metal ion activator, and third one is prosthetic group. So let's discuss one by one, that is coenzymes, what is coenzyme, metal ion activator, and prosthetic group. So the first one, that is your coenzyme. So what is this coenzyme? First one, that is your coenzyme. This coenzyme is a small organic molecule like vitamins and other acting as a if you consider this coenzymes, what is this coenzyme? Some small organic molecule, some small organic molecules, organic molecules, some small organic molecules like vitamins, like vitamins and other acting as a coenzyme, and other acting as a coenzyme. Okay, and other acting as a coenzymes, the other acting as a coenzyme. So it contains small organic molecule and the remaining is called as coenzymes and other acting as a coenzyme. For example, the molecules, the example of this coenzyme, this is your NAD, that is capital NAD. So what is its full form? It is your nicotinamide adenine dinucleotide. Nicotinamide, nicotinamide, nicotinamide adenine, adenine dinucleotide, dinucleotide, dinucleotide. Okay, so it is denoted as NAD positive. Okay, NAD. So this is a coenzyme and it is important in oxidoreductase enzyme. Okay, so this is your coenzyme, this NAD. So it is a combination of vitamins, small organic molecules like vitamins, and remaining coenzymes. So this NAD, so the NAD is an example of coenzyme. This is your nicotinamide adenine dinucleotide, and it is present in the enzymes, that is oxidoreductase. So this is your cofactor or coenzyme. It is found in enzyme, that is your oxidoreductase. Oxidoreductase. So you should keep in mind. So what is the function of this oxidoreductase enzyme? So it involve in the oxidation reduction reaction. So the enzyme is your oxidoreductase, and the non-protein part or the cofactor, that is your coenzyme, is your NAD, nicotinamide adenine dinucleotides. It is a coenzyme and it is important in oxidoreductase enzymes, and it involve in the electron transfer. It involve in electron transfer. One more example is there, that is your FAD, flavin adenine dinucleotides. So another example is there, that is FAD, flavin adenine or adenine dinucleotide, FAD. So what is this FAD? It is a vitamin B2. So this flavin adenine dinucleotide is nothing but your vitamin B2. Okay, vitamin B2, and it is responsible for the conversion of succinic acid to fumaric acid. So what is its role? So conversion of succinic acid, conversion of succinic acid to fumaric acid, conversion of succinic acid into fumaric acid, conversion of succinic acid into fumaric acid. So this is the coenzyme. Okay, so the cofactors are again classified into three types, that is coenzymes, metal ions, and prosthetic group. So this is all about the coenzyme and the metal ion activator. So let us discuss the second, that is metal ion activator. In some cases the cofactor can be a metal ion. So the cofactor or coenzyme, it may be metal ion, metal ion as a cofactor Metal ion has a cofactor. It is known as metal ion activator. Okay, so the enzymes which contain metal ions, it is known as metalloenzymes. So in which the non-protein part is our metal ion. So metal ion act as a cofactor or coenzyme, and this not coenzymes. So it is considered as a cofactor. Metal ion activator as your cofactor, and when metal ion is present as a cofactor, non-protein part, then it is called metalloenzymes, and it contain different metal ions such as bivalent and monovalent. Sodium plus, K plus is there, Mg2 plus is there. So we are very much familiar with these metalloenzymes. Okay, zinc 2 plus is there, and calcium 2 plus is there. So the non-protein part is your metal ion, and it is also act as a cofactor. And the enzymes which contain metal ion, it is known as metalloenzymes. And some enzymes contain the prosthetic groups. Okay, some organic molecules and metal ions are covalently bound to the enzymes for the catalytic activity. It is known as prosthetic groups. So this is all about the chemical nature and catalytic activity of enzyme. In the next lecture we discuss, or we will study, the enzyme substrate complex reaction.